I'm gonna give this until 5 minutes after, and if nobody else shows up, I'm gonna leave this up to you, Nick, if you wanna even like go through this, or if you want me to just post the link. Because you're the only one who's here right now. Yeah, now we've got Destiny, so it's just a matter of like what the two of you want to do. So I know you guys lab partners, so you guys talk, but that's up to you guys. I mean, I don't mind going through this. It's just I don't know if you want to sit here for an hour and a half. So
I'm gonna log into the Kahoot. I'm on my phone. My laptop's in the house. I'm gonna get it. I just got to the house.
sure. I just keep feeling like if I wait a little longer, maybe other people will join, but I don't think they will. I think it's probably going to be just you guys. They might join later on, but yeah, if you guys want, we'll just go through this and stop me if there's anything you want me to explain more, okay? Because since it's you guys, you can be more selfish about it. So let's get going. All right, oh, I can't even read it because all my crap on the screen. Which of the following terms is used to describe removing all microbes, including all vegetative and endospores? All right, so this one, sterilization, pretty straightforward on that one. Just knowing the difference between those terms, mostly memorization for those ones. Right, true or false? Because it involves moisture and heat, boiling is an effective way to sterilize. Of course, so we know adding that pressure to that to help us raise the temperature even more with the autoclave, for example, is what helps us achieve sterilization to remove the endospores, but boiling doesn't remove endospores. How do surfactants work to control microbial growth? All right, yeah, you guys got this. So they have a charged head and a hydrophobic tail, just like our phospholipids do. And they can help basically squeeze in between the phospholipids and break up the membranes, causing the cells to leak open and letting other things get in. Which of the following antibiotics work by affecting the cell wall? All right, so this is going to be penicillins. Basically knowing those bigger ones, if it's on the study sheet, those are the ones you need to know. If it isn't on the study sheet, you don't need to worry about it. So I would know, the main ones for each category, basically. So I know we're going to go over some of the other ones probably in the questions. So I'm going to go over all possible answers there. So which of the following antimicrobials treats helminth infections? All right, so for helminth infections, we're looking at ivermectin here. Ivermectin and praziquantel are like the two most commonly used ones for helminths. Metronidazole is it's a good line of thinking. It is for a eukaryotic infection, but those are mostly going to be used for like amoeba is what they were made for treating amoebic infections, but it's the protozoas. All right, which of the following antibiotics work by affecting protein synthesis? Okay, so for protein synthesis, we're looking at tetracycline. We can look at the aminoglycosides as the protein synthesis affecting ones as well. So it's going to be things like streptomycin and gentamicin and stuff like that. So yeah, but tetracycline and glycylcycline also fall into that category. They're going to affect how the ribosomes function. Let's see if I have more questions on these. I was waiting for it to go on its own. It doesn't do that. Okay. Which of the following antibiotics work by affecting nucleic acid synthesis? Yeah, that's going to be the fluoroquinolones. And then those, of course, end in fluoxacin, like ciprofloxacin. Oh, sorry, I clicked right through that. All right, and if not clicking too much, endotoxin is a pyrogen, true or false? Yep, so this is going to be true. That molecule that's on the outside of gram-negative. I'm getting my emotion alert off. On the outside of the gram-negative outer membrane, lipopolysaccharide, we also talked about it as endotoxin. That is an automatic trigger to your cells that this is bad guy stuff, and also triggers your fever and inflammatory response pretty heavily. So it should not be in your bloodstream. It shouldn't be there at all.
I have a question, Professor Yost.
Yes.
I thought, so pyrogen is like a pyrogen, I thought was like the fever, right? Like the fever.
Mhm. Yeah,
Okay,
anything
so
that causes a fever, yeah.
okay, so endo and exo then, right? Even though exo is the environment outside the cell, or no?
Yes, so if it is exo, anything that comes from outside of the body would be exogenous trigger, an exogenous pyrogen. But endogenous pyrogen can be things like signals sent from your body. So, we call them cytokines. So the cytokine signals can also trigger for you to have a fever response too. So this would be the endogenous ones. But yeah, anything that's not that is exogenous. Those will be the exogenous pyrogens. So yeah, so endotoxin would be an exogenous one. So yeah, sorry for this question that I accidentally clicked it whenever Destiny's asking the question. So phagocytosis refers to a cell engulfing material or other cells outside of it and bringing it inside to be broken down, and that one is true. That's exactly what that means. Cell eating. All right. Which of the following is the most numerous of all white blood cells? Yeah, neutrophils. This is just one you kind of have to know. And if you've seen any CBCs, then you probably already know that one. All right, true or false, macrophages are monocytes that have migrated to tissue. Yes, this one's true. I saw on Instagram, of all places, I saw this little video showing how like macrophages are like basically the ultimate final boss against like, bacteria and other pathogens in the body, because they're the ones that are going to break up and break things down. Even when we have antibody made by the B cells, that antibody isn't doing anything of itself, right? It's just basically a red flag and telling your body that we need to do something about it. And then a lot of times the work that's going to be done against it, a lot of times it's going to be done by the macrophage. It's going to see that the antibody's there and use that as a trigger to attack as well. So yeah, macrophages, man. So they are
Is
going to be
that
in,
the diabetes thing, the
yeah,
diabetes thing? Okay.
yes. So monocytes, whenever they like get a signal, again, when I say signal, I mean a cytokine. So something is produced to call them to the area. They will go through diapedesis and squeeze through the little spaces, if you can even call them that, in between the cells of your, your blood vessels and stuff, and move into the tissues. Once they get into the tissues, they receive other signals, basically activation signals that turns them into macrophages. They'll stay there and proliferate as macrophages in that tissue area. Yeah, monocytes are just kind of like the general ones that are just waiting to get called to a specific area. Right, PAMP. Does that stand for a pathogen associated molecular pattern? All right, yeah, that's true. Just a matter of knowing what it stands for. So pathogen associated molecular pattern, patterns like LPS/endotoxin, and patterns like anything on the surface of a pathogen, or associated with a pathogen, like double-stranded RNA is a good one for viruses. All right, what triggers the classical complement pathway? The classical one. So remember, there's 3 pathways that can trigger complement. Right, yeah, that's his antibody. So lectin is one of the other ones. That's the lectin pathway. And the third one is the alternative pathway. That's going to be typically we're referring to lipopolysaccharide or similar PAMPs like that. All right, what is opsonization? I wish I had a better way for remembering this one. Yeah, so and then when molecules are on the surface of a cell to help the phagocyte eat it up better, like little molecular handles. I like that description. All right. This one could be more than one. What happens when B cells are activated properly? What all will happen? Goes. Okay, so this is actually one more answer to this other than these, but they proliferate and differentiate. That's what they do, right? We activate a B cell, a naive B cell, and the first thing it's going to do is proliferate. Then the proliferated products are going to turn into whatever they need to be. And the 3 things they're going to turn into are going to be plasma cells that is make an antibody. And then we have memory cells that are going to retain memory for the next time we see this thing again. And then the third one is going to be those regulatory cells, which isn't on this list, but that is the other thing they'll turn into. There's always a regulator every time. So no matter what immune cell you're going to be in the acquired immunity. All right, multi-select again. What function or functions does antibody serve? Jesus. It's loud. You guys get to see all these messages that my coworkers are sending me now. Probably they're not going to say anything crazy though. Opsonization, agglutination, complement activation, and I'm just gonna have to. He told me that they have. Okay, whatever. Let me close those. Have that keep popping up. Sorry. What function does antibody serve? So that's going to be opsonization, agglutination, complement activation, antitoxin, and we know we have 2 others. So that's going to be neutralization. So that's where the antibody actually interacts with like the spike proteins of a virus, let's say directly with those proteins to block them from interacting with whatever target they interact with. And the other one is coating. And that's literally just coating the surface of a microbe with antibody, and then it in general cannot connect with anything that it's trying to do. So think of neutralization as being a more specific version of that. Yeah. All right, true or false, cytotoxic T cells are CD8 positive. Yep, so that's gonna be true. Yeah, just a matter of knowing that. So CD8 positive for the cytotoxic T cells, CD4 positive for the helper ones. Those are the only ones you really need to worry about.
Are you gonna put the MH2 and the MH1 or the MHC1 with that or anything like that, or no?
I might, I don't really remember specifically in the questions, but I would be aware of the fact that MHC1 is more associated with the CD8
4.
response. And yeah,
Okay.
MHC1 with the CD8 response, and 4 is the CD4s are going to be kind of involved in both, right? So it just
Okay.
depends on what kind of helper you are. If you are a CD4 cell, you can be a helper on the B cell side or a helper on the cytotoxic side. It just depends on what signals you're getting. So TH1 CD4 positive helper cells, that TH1 is going to be your hint that the TH1 means that's going to be the MHC1, which is the cytotoxic side. And the TH2 helpers are going to be the MHC2 / the B cell response side of things, depending. And of course, I think we've mentioned this before, but just to reiterate, anytime that you're having a response to an infection, it's not going to be just one or the other. Both of them are always going to be happening. It's just a matter of which one's being favored more based on the signals that the body's getting. So if you're infected with the flu, for example, there's flu like outside of the cells too, right? The viruses aren't just inside the cells. So the ones that are outside the cells, when they're picked up by phagocytes or something, then they're going to signal the antibody pathway. So you'll get both going on at the same time, but you just might favor one or the other based on how the body's reacting to it. So yeah, I always kind of think of them as partners. Which immune cells produce granzymes and perforins? All right, so this is going to be a CD8 T cells. That's their weapon, basically, for attacking. When they get activated, they'll get signals from the TH1 helper cells, but whatever, macrophages, and typically what they'll do is, okay, think about it this way. They get activated from maybe coming into contact with your lung cells that have been infected with the flu virus. And so they'll just be chilling, going around, and they'll see that. They'll have to get a second signal from the helper T cell in order to fully activate, because we don't want them just going around killing every lung cell or every body cell or whatever. So they get all these secondary signals. And we often get that signal presentation going on from a macrophage that was in the area first. But if that isn't the case, or a macrophage that was infected, even if that isn't the case, it can happen at the site of the cells themselves. Anyways, once it gets activated, the CD8 positive T cells, now they're going to go over to your infected cells that are infected with the flu, your lung cells, and start creating these granzymes and perforins. The perforins perforate the holes, the granzymes go in and start digesting every content of that cell, and then breaking it down and causing it to undergo apoptosis or cell death. So, yeah. Just eliminates that cell. It's basically like saying, "Oh, there's nothing we can do for you, buddy. We're gonna have to say goodbye." All right, which of the following is an example of a type 1 hypersensitivity? I spend way more time on my work computer than I do on my home computer, so I get confused on how to even use it. Allergy to ragweed. This is going to be the IgE response. Those antibodies involved there. Now we'll remind you guys, when we're talking about the hypersensitivities, those 4 types that we have, type 1, 2, 3, and 4. Remember, type 1 is allergy, we've got IgE. Type 2 is a blood thing. If it has to do with blood, it's going to be type 2, right? So blood rejection of some kind. So it could be you are attacking the wrong blood type that was given to you, or it could be your mother attacking your baby's blood type, right? So that's the hemolytic disease of the newborn. Whatever is going on there, that has to do with antibody as well. It's usually IgM or IgG, and sometimes IgA can be involved in that, just depending on where you're encountering this rogue blood type. So antibodies are involved in that as well. And then when we talk about type 3, the whole point of type 3 is, immune complexes. So guess what? Antibodies are going to be involved in that too. It's usually IgM or IgG that are involved in those complex formations. And then, of course, type 4 is done with the T cells. All the other ones are antibody though. So the delayed type cell mediated is the type 4, that's the T cells. Right. Which of the following is an example of type 2 hypersensitivity? Yeah, so hemolytic disease of the newborn, that's going to be where the O negative mother, who has never seen, quote unquote, seen O positive before, to her, the first time she sees it, it looks foreign, but that's later on in the baby's development. And she's developing antibodies against it for the first time. And there's no evidence that we're attacking it. But the next time you have a baby that is positive, and that positive is your flag for what's foreign, and you've already made antibodies against it, once that baby gets to a stage, that's going to re-trigger creating a whole lot of antibody and having to do with having birth and basically the breakdown of the barriers associated with, the mother to fetus barriers in the placenta. There's breakdown in that whenever we're going to be going through birth. And so that's whenever we see attack of the baby with these antibodies. And there's really nothing we can do if we don't stop it ahead of time. The baby will die. It's terrifying to think of, still happens today. Okay, which of the following is an example of type 3 hypersensitivity? Yeah, so really the only one that we talked about for our purposes in this course was acute post-streptococcal glomerulonephritis. That's a mouthful. But another one that qualifies in here is an autoimmune disease. That would be lupus. I just remembered because that's why I had the freaking wolves on for the question. And I used to teach about how that was one of the examples. There's a lot going on in autoimmune diseases. It's not only going to be like one type of hypersensitivity, but with lupus, we do see antibodies creating those immune complexes and getting stuck in those delicate capillaries like the ones in the face. And then you see the formation of that typical butterfly rash on the face because that's where the inflammation is engaging in those thin capillaries in the face and other areas of the body as well. But and rheumatoid arthritis has a similar effect in the joints. All right, which of the following is an example of type 4 hypersensitivity? Okay, so this one is going to be graft rejection and poison ivy. Both of these are mediated by T cell response. Basically, graft rejection makes sense, right? So seeing somebody else's MHC1, as foreign. So all your cells have MHC1 because any cell can get infected with a virus. And if it has a virus, we want to kill the cell that's infected. And that's the CD8 pathway. So, so you will see somebody else's MHC that isn't even presenting anything, isn't saying, hey, I'm infected with a virus, but it's just chilling. Your body still reacts with that MHC, but sees it as foreign and starts the CD8 pathway, even though there's technically nothing there, but it sees it as foreign anyway. So you start rejecting your graft because of that. And then we have poison ivy, which is basically the oils that were on the leaf of the poison ivy plant interacting with your skin in a way that it looks like it's part of your cells, and so your body will interact with it that way. So. And SCID is the primary hyposensitivity, genetically related. Okay, so you run an ELISA to check if your patient has antibodies in their serum against RSV. What kind of test is this? Oh, sorry, I just saw that there's chat. Yeah, sorry. All right. Yeah, so this is indirect. Indirect is when you're checking to see if the patient has the antibody, and the direct is going to be seeing if the patient has the antigen. Again, sandwiches go directly in my mouth. That's how I start building this thought process in my mind. When I haven't taught this in a while, like literally when I'm doing the traditional classes, and then we get to ELISA and immune system and stuff, I'll have to like remind myself, like, okay, how do I remember this? And because I don't, doesn't not come naturally to me. I have to think through these processes every time. But I love immunology, but man, it is a lot. All right, could be more than one here. Which of the following are respiratory diseases? Okay, so we've got pertussis and tuberculosis for this one. Remember, gas gangrene is going to be skin related. We have syphilis, that's going to be sexually transmitted. We have smallpox, that's skin, and then cholera, which is a GI disease. I'm probably going to give away answers later, but that's okay. What type of illness is caused by Yersinia pestis? I don't say it enough. All right, so that's gonna be the plague, and the reason that the picture's up there, I used to have people like select the like reservoir for the plague that we have here in the United States. And of course, the reservoir in the United States for the plague is prairie dog. I mean, so I used to live in Yukon. I'm on this group called Yukon Happenings in Facebook, and I swear these people want to save every prairie dog in Yukon every time a new building goes up. And I think that's cute, but like I'm also at the same time like they do carry disease. So I don't think any of the prairie dogs in Oklahoma have tested positive for it, but you know, it can jump at any point in time, and it will if we see continued urbanization. So, all right, what is the most common infectious disease in humans? Yeah, this one was a shocker for a lot of people, but it's just dental caries. So tooth decay. Not a surprise when I say it to people, really, but nobody would ever think of it to answer that question if they didn't already hear me say that. All right, what causes river blindness? So we talked about two different forms of blindness when we talked about the skin and eye diseases. Which one causes river blindness? Okay, so river blindness is actually caused by a helminth specifically. So it's going to be a worm that makes its way into the eye and causes blindness there, usually from the inflammation associated with the worm. Anyways, interesting fact about that worm is that it has a bacteria that it is infected with called Wolbachia. And that Wolbachia bacteria inside of the worm is what actually makes it pathogenic. It gives us its pathogenic traits. And this is true for a lot of helminths. They have that similar bacteria. But recently they've discovered if they introduce Wolbachia, that bacteria that makes the worms pathogenic, they introduce that into a malaria situation with the protozoa that cause malaria, that it actually causes the malaria to be unable to cause disease. And so they're looking into that as a potential way to control malaria infection. I think that's super cool. But yeah, so river blindness, that's the helminth that is, oh gosh, it starts with the O. I can't remember it because of course I'm thinking about freaking malaria. Onchocerciasis, I believe. And yeah, that's right, onchocerciasis. And then the other blindness we were talking about, the most common infectious blindness in the whole world, that of course is caused by chlamydia trachomatis, same thing that causes chlamydia in sexually transmitted diseases. All right, true or false, endotoxin is part of gram-negative bacteria outer membrane. I already talked about that one. Okay, I'm not going to hold it against you if you don't get it. So this one is true. So the endotoxin is part of, because it's part of the bacteria is why it's called endo, because endo to the bacteria, right? Bacteria, gram-negative bacteria on their surface, that lipopolysaccharide is very unique to them, but it's a signal to our body that it's bad guy. So that's why it's a toxin, but that's on the outside of the membrane of the gram-negatives. We talk about it a lot. So what is a common cause of vaginitis? Remember what we said about itis? Alrighty, so Candida albicans is yeast, so that image was actually slowly building up to show us budding yeast. So vaginitis, remember that's when we're going to have actual inflammation of the vaginal canal. We're going to see that with Candida and with Trichomonas. Other ones cause pretty serious inflammation of the, reproductive tract, but not the vagina. All right, Lyme disease is caused by the organism Rickettsia rickettsii. Is that true or false? In my unit 2, I think, no, unit 3, Kahoot thing that you can go through, it's like constantly asking, like, is this disease caused by this? And they'll just like switch until like it comes to the right one. And that's a good way of testing your memorization of like what causes what, basically, especially when it comes to like tick-borne diseases. This is false. Lyme disease is caused by Borrelia burgdorferi. And Rickettsia rickettsii does cause a tick-borne disease. That's going to be Rocky Mountain spotted fever. Right, Chagas disease is caused by the organism Rickettsia rickettsii. Guess I brought a lot of those questions over here. Oh, the microphone. All right, so that's false, right? Rickettsia is Rocky Mountain spotted fever. Chagas disease is Trypanosoma cruzi. There we go. Rocky Mountain spotted fever is caused by Rickettsia rickettsii. I forgot how many of them I put over here in this review. Jeez. All right, Chagas disease is caused by Trypanosoma cruzi. I did just say it, but whatever. This is the bug. That's the bug that spreads it, by the way. The triatomine bug or the kissing bug. It bites and then it defecates, and when it defecates, the protozoa make its way into the skin and cause an inflamed area called the chagoma. All right, Lyme disease is caused by the organism Borrelia burgdorferi. Is that what I just said? Yes, it is. Man, I should have just let it play out on its own. I'm really bad about that. All right. Could be more than one. Which of the following diseases are hemorrhagic fevers? Okay, so just loss of fever and yellow fever for this one. Rocky Mountain spotted fever is not a true hemorrhagic fever. You do get spots from some petechiae involved in that, but because we don't have that form in the same way that what we would see with loss of fever and yellow fever, it doesn't really count. Cat scratch fever and trench fever. Man, I mean, they're not something that you're going to see a whole lot. With cat scratch fever, you're going to see, your lymph nodes swelling, creation of buboes, right? It is just like buboes, like what you would see with the plague. That swollen lymph nodes are actually infected with the bacteria. With trench fever, this is like what we see with the homeless folks. It's transmitted by lice bites, and then ehrlichiosis is a tick-borne disease. And it's not super serious. It's a bacterial. All right, true or false, septicemia refers to when bacteria are flourishing in the bloodstream. Is that the correct term? Right, so this is a issue that a lot of people sometimes have with the terminology. When we say bacteremia, means you have bacteria in the blood. There shouldn't be any in there, right? There should be no bacteria in your blood at all. If there are bacteria at all, that's bacteremia. We would be concerned about how they got in there. But if they're flourishing and actively infecting your bloodstream and growing in there, that's septicemia. That's when you become septic. All right, true or false, keratin is the waterproofing protein on the skin. Easy one, yeah, that's true. It's also part of what makes the outer coating of endospores. All right, so what does lysozyme do? This is part of the innate response. Yeah, so the lysozyme is an enzyme that breaks down peptidoglycan. It's a very innate type of response. It's so general. It will defend against pretty much anything that has peptidoglycan. It's going to be present in like your tears and saliva and stuff. All right, multi-select again. So what is Cryptococcus neoformans? All right, stretching the understanding basically of what is, causing what and what they're called and everything. So Cryptococcus neoformans is a fungus. The reason I had the pigeons in the picture, it is spread in pigeon feces. It is a cause of meningitis. More commonly, we see this in the immunocompromised homeless population. A lot of times they're going to be HIV positive. Right, what is the causative agent of whooping cough? All right, so this is just a matter of memorizing these guys. It's going to be Bordetella pertussis. There's another Bordetella out there, which you will, if you have dogs, and you will probably know, but another Bordetella bacteria that causes cough, kennel cough in dogs. So you'll have to get your Bordetella vaccine for your dogs to prevent their coughing. I always kind of remember that. I don't know why I associate kennel cough first, but whatever. What causes primary amoebic meningoencephalitis? So we really only talked about two causes of meningoencephalitis. All right, so PAM, or primary amoebic meningoencephalitis, is caused by the amoeba Naegleria fowleri. So meningoencephalitis always going to be amoebas. Primary versus G, which is granulomatous. Okay, so that's a good question, Destiny, about the difference between the Tdap and the DTaP. And it has to do with basically how the antigens and the toxins are associated with carrier proteins and at which stage you're in as far as the order of vaccines, if you're an adult getting the vaccine versus getting your first shots as a baby and stuff like that. Other than that, I don't know a whole lot about it, but there is a difference. Yeah, there's a reason they refer to it differently, right? But yeah, but other than that, that's all I know about that part, is basically what stage you're going to be in. And I'm sure you guys know, like, now that you've learned about immunology and you kind of start to understand like how your immune system responds. When we talk about the COVID vaccine, like if you got this type of vaccine, like let's say Johnson and Johnson, and they would write that on your card, they want to be sure that when you come back for your booster, you're getting Johnson and Johnson again, and you're not getting one of the other ones. That's just one example, because one type of COVID vaccine has the mRNA version, which literally just mRNA in a little vesicle that they put into your body, and your body starts reacting to the mRNA, and your cells will produce the protein, and then your body reacts to that and creates antibodies against it that way. Or they have the viral version, which is a adenovirus, which just normally causes cold in people, but they've cleared it out and put some genes for the protein for COVID so that they have spikes from COVID. And so when the body sees those spikes, they react to it the same way that they would if they saw COVID with those spikes, the same spikes. But you don't want to mix up those vaccines, right? If you got the mRNA, you want to be sure your boosters have the mRNA. If you got the virus, you want to be sure that your boosters have the virus. And so that's why they keep such close, survey of that whenever you're getting your first shot and your second shot and so on and so forth. But yeah, so that's why they do that way. And it's sort of a similar thing of what's going on with Tdap and DTaP, which just has to do with how those toxins, because they're all toxins that are in there. So we're talking about tetanus, diphtheria, pertussis, right? Or diphtheria, tetanus, pertussis. But it really has to do with how they're interacting, like how they're associated with one another, because they're all haptens. They're all too tiny for the immune system to react to. So how they're associating with each other and what kind of carrier, and also what kind of adjuvants are going to be added in there. Remember, adjuvants are added in there to provoke an appropriate immune response or control it in a specific way. So it can get complicated, but that's as much as I know about the difference in them. If you look into it more, if you're interested, and I might look into it, I just never have gotten around to it. Let me know what you learn. But yeah, so the amoebas are either going to be primary or granulomatous, so PAM or GAM. And PAM is Naegleria fowleri amoeba, and then GAM is acanthamoeba. And it's just whatever is causing it for you. They're both just as deadly. They're both like 98% fatality rate. There's not a whole lot we can do for people. Now, what is the defining stain used to identify the bacteria that cause tuberculosis? Boy, if we don't know that by now. What stain is important for the bacteria causes tuberculosis? And this is an image of that stain that's coming up. All right, so this is the acid-fast stain. Remember, it's acid-fast because the bacteria, Mycobacterium, it could be Mycobacterium tuberculosis or Mycobacterium leprae or Mycobacterium whatever. But these guys are acid-fast, meaning that if we stain them with carbofuscin, for example, which is what we did in class, then rinse them with acid, they're going to hold fast onto that carbofuscin stain because the stain is lipid soluble and it's not going to care about the hydrophilic properties of the acid or the alcohol. So it's going to hold tight onto the lipid, the waxy surface of Mycobacterium, which is mycolic acid, but the proper name for that stain is acid-fast stain. That waxy coating that they have is why they're so resistant. All right, HIV codes for something called reverse transcriptase. What does that do? Right. So reverse transcriptase. So think about it this way. When we learn about DNA and RNA and protein, right? So we learn DNA to DNA. If we're making copies of cells, we make DNA copies. That's going to be replication. But DNA holds genes, and the whole point of the genes is to make a protein. And in order to get there, you need to make a copy of that gene. That's your working copy. That's your mRNA or messenger RNA. So we are writing our nucleotide code from DNA into a different nucleotide code in RNA, but it's still nucleotide. So we're just transcribing, writing over, like a scribe would do, a copy of that gene, taking that copy outside of the nucleus Smithsonian Institution, like I like to say. And now you can work with it outside of the nucleus with your ribosome on your endoplasmic reticulum, the rough ER. And that ribosome will read that messenger, that message in the messenger RNA. And that transcript messenger RNA will read it and will spit out amino acids appropriately according to what codon codes for amino acid, amino acid, amino acid. And that sequence of amino acids, that primary structure of your protein, we're translating then, because that's a different language now. We're not going from nucleic to nucleic, we're going from nucleic to amino acid, different language. So that's translating to a different language, right? So that's how I remember DNA is just replicating, making a copy. DNA to RNA, we're transcribing, it's still the same language. And then RNA to protein is translating. So it's different language. So anyways, reverse transcriptase has the answer to the question in it. So reverse transcriptase, ACE means it's an enzyme. You know that already, just by the ACE. So transcript, we know that that is what you made when you went from DNA to RNA. So your mRNA is called your transcript. That's what you transcribed from your DNA. So if we're going in reverse, we're going from RNA into DNA. So that's where we get that answer from. Yeah, so HIV has a single-stranded RNA genome, and one of the first things it does with its little reverse transcriptase, which it will package in with the viral particle, is turn it into double-stranded DNA, which can then be incorporated into your genome or genome of whatever cell it's infected. All right, what is the causative agent of chickenpox? Yeah, so that's going to be varicella zoster. If you guys didn't hear my story about Edward Jenner and how he discovered that, cowpox could be used to be a vaccine against smallpox, different viruses, the vaccinia virus versus the variola virus. But vaccinia vacca for cow, right, is what we named vaccines after because it was the very first vaccine. Y'all, he tried this whole thing out very first time ever with his freaking son. He inoculated his son with vaccinia virus and then pushed him out into a society that had, smallpox, which is a deadly disease. Son was fine. He proved his point, but geez, man, maybe pick someone you don't know. All right, smallpox has been eradicated from the planet thanks to widespread vaccination. Is that true? Has it been eradicated? This person is pointing at a scar on their arm. That is exactly what type of scar we will see form from smallpox vaccination. So a lot of times, whenever we see bovine products present in vaccines, so it originally was associated in the case of creating the vaccine for smallpox, yes, but we don't vaccinate for smallpox anymore. So because it's been eradicated. So yes, it has been eradicated. But sometimes we see products from bovine specifically because they can relate pretty closely to the human products, and it's easier to manipulate tissue samples from cows as opposed to getting tissue samples from people in order to do the testing, if that makes sense. So bovine, or you might see bovine, or you see porcine, right? So pork, that's going to be pigs, right? They're both pretty close to some of the human proteins of what they're going to be using for studying for these vaccines. So you'll either see typically one or the other a lot of times if we can't work with human tissue. We do work with human cells whenever we were making the COVID vaccines. They did use, the human embryonic HEK293 T cells, and they're human embryonic kidney cells from already disposed of embryos. There were no embryos that were killed in order to make these vaccines. And people think because it's embryo, well, you would need to eventually. No, I promise you, like we've had lines of these cells going on since like at least the 60s that we've kept alive for like forever. I don't know how they got them back then, and that's not my question, but I know that if they already did it, why would I waste, this wonderful thing that has helped us study medicine in humans that keeps other people from being killed and other people from getting sick and studying genetic diseases. So we already did this bad work, and maybe that's bad, and we're not going to do it anymore. But I'm not going to throw away this stuff that people already, established. So it's not hurting anybody now. So that's the idea of it. Doesn't mean to venerate it. It just means that we should use it if it is a tool that is available. We're talking about deadly diseases, right? So that's the idea. You can argue with it however you like, but that's the concept. So with smallpox, it has been eradicated. We had widespread vaccination, again, with the vaccinia virus, create a little scarring on the skin, and the body would react to it and heal it. And when it did that, we knew you had appropriate antibodies against it. The United States no longer vaccinates against smallpox because we haven't seen a single case of smallpox in the entire world since like the 80s, at least, I think. So we don't have it, but we have cold storage that we know of in several countries United States has at least one cold storage at the CDC, and Russia has cold storage. I think there's other countries, probably China, but I don't know. I'm assuming, right? So it still exists on the planet, but it isn't actually causing disease anymore. The whole problem is they're worried about it being used as a bioterrorism agent, which is entirely possible. Russia's prepared for that. They still vaccinate every single citizen. I had a student who was like 19-year-old who had been adopted from Russia, and she had already had vaccination against smallpox. But I will warn you, no matter what the boomers say, no matter what other people might say, vaccination against smallpox really only lasts about 10 years. After that, you have to have boosters. So yeah, my mom is all like, "You got this scar. I'm not going to get the smallpox." And I'm like, well, I hate to break it to you. Ain't doing you good now. But yeah, so smallpox, very serious disease, actually extremely deadly, very disfiguring. And it has been eradicated thanks to vaccination from Edward Jenner being a weirdo with his kid. And, now we don't even vaccinate for it anymore. All right, so what organism causes syphilis? When you think about like whenever, like the Spanish or whoever would be like sailing across to conquer new lands, and they would give natives of people like blankets with smallpox, literally, in order to kill them. Yeah, here's your blanket. Sure, you're welcome. Wink, wink. Just people haven't changed a whole lot, right? All right, so bacteria. Yeah, we're just going to ask what bacteria. Next question. Okay, in which phase of syphilis does the canker or whatever, I can never say the word appropriately, appear? I think it was like somebody told me it was like chancre. It's not canker. I want to say canker, like a canker sore, but it's not how we say it apparently. All right, we see canker, or we must call it canker because that's how I want to say it. We see these sores develop in the primary phase of syphilis. Primary phase is that one single canker sore. It's a hard raised area where you have the syphilis bacteria entering into the body. Secondary, we have development of that characteristic brown rash. It could be all over the body, but it's primarily seen on the palms of the hands and the soles of the feet. There's other like typical, like you're sick, like lymphadenopathy and sort of those sorts of symptoms there as well. Then the tertiary phase is whenever you have development of, what I want to say, neurological symptoms like dementia and even loss of sight and eventual death associated with it. And there's not a quaternary for this one, it's just 3. And the bacterium, in case it doesn't bring it up, let's see, is Treponema pallidum for that one. All right, so what is pharyngitis? Yeah, inflammation of the throat. The pharynx is your throat. Itis, of course, is inflammation. Which of the organisms below is a helminth? Gross. All right, great job. This is going to be Ascaris lumbricoides and Schistosoma masoni. I think it's Mansoni. I think I just spelled that one. But anyways, those guys are the true helminths. The Plasmodium and the Giardia, those guys are protozoans. They are both eukaryotes. Everything on here is a eukaryote, but just don't confuse the helminths with the protozoans. They are different. Helminths are multicellular animals, technically. Right, which diseases are carried by viruses, or caused, excuse me, by viruses? Some of these are bacteria, some of these are protozoa. All right, so as a matter of kind of, flashcard time for these guys, right? So rabies is a virus, chikungunya is a virus, and dengue fever is caused by a virus. Whooping cough is a bacteria. Toxoplasmosis is Toxoplasma gondii. That's the one that's in the kitty litter. That's dangerous for pregnant women. Lead to miscarriage, so please don't ignore that. So that one is a protozoa. And gonorrhea is Neisseria gonorrhoeae. That is going to be bacterium. That might sound familiar. Neisseria gonorrhoeae, gram-negative Diplococcus, just like Neisseria meningitis, gram-negative Diplococcus, same genus. So Neisseria is kind of that one, but yeah. So that's great. We did it. So here is your podium. You guys have any other questions for me?
No, ma'am.
All righty. Thanks, guys, for hanging out with me today. I kept it a little shorter. And good luck studying. And you guys can always email me if you need anything while you're studying. And I will see you guys on Monday.